So after discussing the partial hemolytic organism, now discuss the complete hemolytic. So first algorithm remains the same. Gram we are discussing gram positive, catalase negative, straps, and now we do hemolysis, and in the hemolysis we have the complete hemolytic green in color bacteria. So we have two groups. First one is group A and second one is group B. So we have strep pyogenes, streptococcal pyogenes or pyogenes. So this is complete hemolytic strep, and how to diagnose it? We do after hemolysis we do pacitration test, and also we do PYR status. So strep pyogenes is bacitration sensitive and PYR positive, and the strep agalactia is bacitration resistant and PYY negative. So strep pyogenes is having the first important exotoxin is erythrogenic toxin, and this toxin is also known as toxic shock like syndrome causing toxin, which again cross-link TCR with the MHC2 and causing the cascade of cytokines release of the cytokine cascade, leading to fever, which is known as scarlet fever, along with rash. These rash are sandpaper like rash on the whole body. Then we have the circumoral pallor, and also we have the strawberry tongue, and this erythrogenic toxin also causes pharyngitis. So we have a pretty much common case scenario of strep pyogenes infection in a child, and in this scenario a person is having pharyngitis, fever, and after some day having a rash, circumoral polarity, and strawberry tongue. So after causing this toxin causing scarlet fever, the next important virulence factor of strep pyogenes is its hyaluronic acid capsule and M proteins, and these two important in order to evade immune defense from our body. So specifically the M protein, after causing pharyngitis, it has the molecular mimicry type of mechanism in which the M protein they undergo molecular mimicry in which they mimic like the molecules, especially the epitopes, and releases the cytotoxic cells to causes the human cellular myosin and tropomyosin protein and and causes heart problems, specifically rheumatic fever or rheumatic heart disease. So rheumatic fever is because of this strep pyogenes strep M protein. So in the rheumatic fever scenario, we have heard that the patient is suffering from impetigo a week ago, and after fortnight, or he's suffering from any kind of heart problem like murmur or any kind of thing. So the next important infection is impetigo, but this impetigo, impetigo is superficial infection of the epidermis, but it is the second most common. First most common is Staph aureus. Second most common cause of impetigo, having the honey crust like lesions. And the strain which causes impetigo also causes glomerulonephritis. So in the scenario of post streptococcal glomerulonephritis, the patient is having some kind of sore throat or impetigo. Both, mostly the scenario gives the clue of sore throat, but occasionally, or whenever the examiner try to trick you, then he give that impetigo type of clue, that the patient is previously suffering from impetigo and now present with this dark urine presentation. So that clue is very much important in order to accurately diagnose either it is glomerulonephritis or any other nephritic or nephrotic syndrome. So it is also causing impetigo, also causing non-purulent cellulitis, which is also epidermal infection cellulitis, dermal infection cellulitis, but it is non-purulent. Purulent is Staph aureus and non-purulent is Strep pyogenes. Then it is also causing necrotizing fasciitis and eryplasis, uh, eryplasis and necrotizing fasciitis. And after these two or four important conditions, now we discuss how to diagnose or how it causes a different type of there is one more thing, that in children it causes it is the second most common cause of osteomyelitis. So now how to diagnose this? We can do a throat swab in a general OPD setting. Throat swab detecting the rapid antigen test. Throat swab for rapid antigen testing in order to diagnose this. And also we do ASO titers and anti-DNase B titers. So what is ASO? Actually, there is a streptolysin toxin O, which is RBC membrane lyser, and if the ASO titers along with DNase B is high, then it is depict that there is a recent infection of rheumatic fever, either rheumatic fever or glomerulonephritis. So this is the brief discussion of strep pyogenes. Most important, I have mentioned most important clues which I experienced most high yield and most favorite for the examiner. From the examiner point of view, I have discussed this strep pyogenes. The next one is, again we are discussing gram positive, catalase negative, which means these are streps. We have discussed hemolysis, so these are complete hemolytic or beta hemolytic. So now the differentiating test is bacitracin. So we have discussed bacitracin sensitive organism. Now we are discussing the bacitracin resistant and PYR negative negative test bacteria. So this one is strep agalactiae, and strep agalactiae, pretty much the normal flora of it colonizes in the GI tract and genitourinary tract, especially the vagina of the pregnant, especially the pregnant female. So over there it colonizes and it can lead to strep infection, especially if the pregnant female is unvaccinated. So there is no protection for the neonate while passing through the birthing canal. It acquire infection and causes neonatal. First one, neonatal first most common cause of neonatal meningitis. Also it can cause neonatal sepsis or neonatal pneumonia and soft tissue infection. So in order to prevent this, we always there is a universal screening of all the pregnant females which are at their 35 to 37 week of gestation. If they are positive, we give them intrapartum ampicillin and penicillin in order to prevent this neonatal meningitis and neonatal sepsis. And also it is also releases it is having post-streptococcal polysaccharide capsule, which make it this organism include in the list of our mnemonic please shine my skis, and which are causing the asplenic or hyposplenic patient, causing them infected. And also it releases hippuric acid or hippuric acid or hippuric test is positive. And also it releases camp factor. This camp factor is again RBC lyser, making them beta hemolytic or complete lytic species. So this is the discussion of strep agalactae. We have discussed the strep beta hemolytic organism. So this is all about strep pyogenes and strep agalactae.